Is my screen visible now in presentation mode?
Yes, sir.
Good morning, all of you. And last class we discussed how to conduct a BOD experiment. Okay, so BOD we are already familiar. You have to estimate the oxygen demand, and that the organisms or microorganisms in the wastewater will consume organic matter as a food, and it will also exert BOD in terms of milligram per liter. Okay, so that is very simple. You just find out the DO of the sample initial, that is day 1, and incubate at 5 days. That at 20 degrees Celsius. After 5 days, you just take out the sample, measure the remaining DO, initial minus final DO will give you a BOD of the sample. That's what we discussed. Okay, and if you keep within 5 days, and it is possible to that either consume or oxidize only the organic fractions in the carbonaceous matter, because organic matter contains carbonaceous and nitrogenous matter, and if you do only within 5 days of incubation, you could able to convert carbonaceous material into carbon dioxide. So that only that this is BOD 5. That's what we have discussed. If you go beyond 5 days, there could be a possibility of conversion of nitrogen contents into other products. That's what we have discussed. That is nitrite and also nitrate by Nitrosomonas and Nitrobacter bacteria. And either that you can call it as nitrogenous BOD, or you can call it as simply as ultimate BOD. Okay, these points were discussed. Okay, CBOD, you are familiar, carbonaceous BOD, and nitrogenous BOD, and BOD ultimate we have discussed, and you can also have a total BOD. Total BOD in the sense is carbonaceous plus nitrogenous matter. Okay, yes, yeah. So we'll move on to nitrification, and imagine, and if you are now prolong your BOD experiments beyond 5 days or after 5 days. Okay, there could be a sudden other group of bacteria, that is nitrogen consuming bacteria, Nitromonas or Nitrobacter, or otherwise is nitrifiers. So you can make a note on nitrifiers. Now will feed on like the nitrogen compounds or nitrogenous material in the wastewater and convert into a nitrate. Okay, so nitrogen into nitrate is not a single step process, it's a multi-step process, two-step process. So protein what you consume, and that may contain a lot of organic fractions of organic nitrogen and hydrolyze into free ammonia and NH4 plus ion. These free ammonia and NH4 plus ions are converted into nitrite, that is by Nitrosomonas, and nitrite to nitrate by Nitrobacter bacteria. So thereby the wastewater contains rich amount of nitrate. Okay, that's what has been mentioned. Okay? So this is something is important, and you can draw a graph in between time of incubation versus the oxygen consumption in terms of milligram per liter. Okay, so this may not have a straight line. Please make a note on this, may not have a straight line. So this is some like shape. So this normally they may call it as is a lag phase. Okay, bacteria present in the BOD bottle may not consume the organic matter very immediate, because it will understand the system, it will understand the surrounding environment, then only it will start grow on, it will consume oxygen as a or consume organic matter. Well, degradation of the organic matter will also consume oxygen. Okay, so please make a note on, it's not like a smooth graph or curve like this, and you may give a graph like this. Okay, like this. Okay, so this is up to 5 days. That's what is called as the carbonaceous BOD, and beyond 5 days, or you can have, because here maybe 5 days. Okay, and beyond 5 days there could be a increase. Okay, in the oxygen demand or oxygen exertion, that is because of nitrogenous matter, and if you add all together, that's what I told you, total BOD, TBOD. Okay, carbonaceous BOD and nitrogenous BOD are too familiar. Okay, this is simple. You can call exertion curve, BOD exertion curve. Okay, yes. Okay, this is what I told you. Two different bacteria are responsible. These are autotrophic bacteria, Nitrosomonas and also Nitrobacter. This story I have already mentioned to you, because is the protein, whatever we consume, protein contains 16% of nitrogen. Okay, and this is entirely on the organic nitrogen. So organic nitrogen definitely or undergoes a hydrolysis. It may converted into like ammonia. Okay, so this again, this nitrosomonas consumes this ammonia, and it may also go for these sort of equations. Please make a note on this. Something is important. Otherwise, you just remember simply. That's what I told you. Protein content contain organic nitrogen. Organic nitrogen will go hydrolysis, and it will form NH3 and NH4 plus. NH4 plus. This NH4 plus is taken up by nitrosomonas bacteria, which will convert everything into nitrite. Okay, make it very clear. So at least you remember this equation, how the conversion of ammonium ions into nitrite. This is by nitrosomonas bacteria. Okay, yes. And second one, you may get nitrite. So all nitrates will be converted to nitrates. Nitrites be converted into nitrates by Nitrobacter bacteria. Okay, so nitrite to nitrate. Okay, so as a whole, at least you remember how the conversion of ammonium ion into nitrate in a single reaction. That's what it is being. Just refer the notes later, and if you add equation number 3, that what we have discussed in the previous slide, and equation number 6, this slide, this one. Okay, and if you add or combine together, and you may get a equation like this. That this is how the conversion of ammonium ion into nitrate by a single. So this is enough for the examination point of view. So please remember these concepts very nicely. Wastewater contains a lot of protein or whatever we consume as a protein products. Okay, and the wastewater also contain large amount of organic nitrogen. Organic nitrogen undergoes a hydrolysis process, which may be converted into ammonia, free ammonia, and NH4 plus ion. Okay, this NH4 plus ions will be again taken up by one group of bacteria called nitrosomonas, which will be converted into nitrite. Okay, and byproduct of the first stage will be taken up by the second group of bacteria called Nitrobacter, which will convert everything into a nitrate. Okay, yes. So this is one simple equation you can remember, how the conversion of ammonium ion into nitrate in the BOD reaction or during the degradation of the organic matter. Okay, yes, yeah. We'll move on to the second one. Okay, you can also measure the organic content in terms of chemical oxygen demand. Please make a note on COD, chemical oxygen demand also. You can also measure in terms of chemical oxygen demand. Okay, so BOD you are already familiar, the oxygen demand by the microorganisms. Okay, now what you do is very simple. You can use some strong oxidizing agents. Thereby, you can convert the organic fractions into a oxidized product. It's very simple. So please remember that a simple equation called like this. Okay, so we have organic matter. Okay, and potassium chromate is the strong oxidizing medium. These reactions may happen in a low pH range in acidic medium. And you can also add catalyst. Okay, and this to speed up or to digest the solution, you have to heat the sample. Thereby, one product is getting oxidized and other is getting reduced. Okay, chromium is getting reduced and organic matter is getting oxidized into carbon dioxide. Okay, so please remember this is something very important. Okay, COD is very simple. That this is also again an oxygen equivalent. By using a chemical. Please make a note on this point, is important. Chemical to oxidize the organic content of the waste. That's what I told you. Just use strong oxidizing agents, and thus this oxidizing agents will be used to convert the organic matter into an oxidized product. Okay, so here in the COD experiment, they may use potassium dichromate as a oxidizing medium, but it will work out efficiently in acidic medium. So please make a note on potassium dichromate is an oxidizing agent. It will also work out in an acidic medium. So this test will be conducted or must be conducted at an elevated temperature. And to speed up the reaction, you can use catalyst. Okay, in the COD reaction, you may use silver sulfate as a catalyst. So please remember chemical to oxidize, and even organic matters are getting oxidized into carbon dioxide. Your oxidizing medium is that potassium dichromate, and it will work out better in acidic medium. And this test must be conducted at elevated temperature, and you have to also use some catalyst, silver sulfate. Okay, silver sulfate may not participate in the reaction, but at the same time it will also speed up the reaction. That's what is mentioned in the last point. To aid in the oxidation process, and you may use silver sulfate as a catalyst. Okay, yes. And this is what I have mentioned: organic matter in potassium dichromate in a low pH condition, that's an acidic medium. You can use silver sulfate as a catalyst at elevated temperature, and chromium is getting reduced and organic matter is getting oxidized. So please make a note on. So chromium, initially, if you calculate the oxidation state of Cr2 O72 minus, you may get +6. Oxidation state is +6 and chromium 6 is +3. Thereby, chromium is getting reduced and organic matter is getting oxidized. So that you just remember in this case. Okay, and this you can do it in two different methods. Okay, you can estimate the COD of the sample in COD of the sample in two different methods. One is about open reflex method, another is about closed reflex method. It's very simple, and what you do is you have a apparatus like this. Okay, apparatus like this. You could be able to see now round bottom flask. Okay, and you can also take up the sample with dilution or without dilution. You can add potassium dichromate. You can also add sulfuric acid because it is acidic medium. You can also add, say example, silver sulfate, and you can heat the sample in an elevated temperature, and there could be a evaporation of the sample, and you can also have a condensation mechanisms. You are not in a position to left out the vapor. Okay, so there could be a condensation mechanism, something like the water will be circulated thereby, and the vapor is getting further condensed and it will also leave out in the same system. Okay, so after digestion, what do you do? Just take out or transfer this sample in the conical flask, and you just titrate against ferrous ammonium sulfate using ferrion as indicator. It's very okay. Please remember. Okay, take out the sample, add potassium dichromate, then make it in acidic medium, and add sulfuric acid, and also add silver sulfate as a catalyst, and heat it at elevated temperature. And after digestion, okay, because heating at elevated temperature, you can simply call it as digestion. Okay, digestion, and then at a particular temperature and particular duration, and there could be and you could able to see now the water circulations, and thereby condensation also may happen, and volume may not change. After digestion. Take out the sample and transfer from this round bottom flask into a conical flask, and burette solution is ferrous ammonium sulfate, and indicator may be a ferrion as indicator, and maybe titrate against and just find out how much amount of the volume consumed in terms of FAS, and you have a standard calculations, and based on that you can calculate what is COD of the sample. Okay, and you can also do methods like closed reflex method. Okay, because it is a time consuming process. Okay, and also you may need a bigger volume of the sample, or you need to have larger volume of the sample, to demonstrate these sort of experiments. At the time, you may also go for at a max 6 samples. And if you want to measure 20, 25 or 30 samples as a single time, it is very difficult. Okay, 6 at a time, and you have to repeat for 5 times. So 5 into 2 per 10, almost 10 hours may take to estimate the COD of the sample. So now in the laboratories, and people have used closed reflex method. Closed reflex method is very simple. Okay, you could able to see like a vial. Okay, small vial. So in the vial, you just add sample and dichromate oxidant plus sulfuric acid plus silver sulfate as a catalyst. So, and prepare the sample and keep it inside the digester. And adjust the temperature and your time. Switch on the instrument after digestion. What do you do? And you just take out this vial, and keep it in the spectrophotometer. And you may measure for the absorbance. Is it fine? You make a difference on what is this closed reflex method and what is open reflex method. Is it okay?
Sir, but absorbance value we may get due to other constituents also, no, sir, in the sample.
Because this is clear, no? This is very clear solution. Because anyways, the water with the organic matter, this is a clear solution. And if you add the potassium dichromate, this is yellow in color, and it may represents only one particular absorbance. Depending upon your organic strength, there could be a changes in the absorbance value.
Okay, sir.
Yeah. Okay. So this is what I told you, and you just take out the sample and add sulfuric acid and potassium dichromate, and just digest the sample, and ferrous ammonium sulfate as a titration. And then here in the closed reflex method, and you may also do a digester vial and COD just measure digest in a COD digester and take out these vial after digestion and keep it in the spectrophotometer. You just measure absorbance and related to COD of the sample. So this is what we have discussed as a theory. So for examination also, just your logical understanding is more than enough. Either you just represent as a you need not in a position when you need not write a text as a paragraph voice. Based on your understanding level, you can also brief out like this way, or you can also brief out something like a flow chart or something. Okay. Yes. Yeah. So being familiar with organic matter, biological, say predominantly biological characteristics, we'll move on to now the microbial characteristics of the water, microbial characteristics of the water. Okay, yes. The principal microorganisms of concern in the water quality are bacteria, protozoa, virus, helminths, and also algae. So please make a note on this. So we are going to discuss bacteria, protozoa, virus, helminths, and also algae. So bacterial infection you would have come across, protozoa infections, viral infections in the water, helminths eggs, and also algae. So we'll see one by one. And bacteria, these are single cell microorganisms you would have already come across, and the ranging from 0.1 to 10 micrometer level. Okay, size of the bacteria in water in the range of 0.1 to 10 micrometer, and is relatively very simple structure. This is easy to identify by normal microscopes. Okay, and this bacteria contain one cell membrane, and then bacteria contains cytoplasm and also nucleus. This contains bacterial interior contains is two regions. Main region is cytoplasm and also nucleus, and but both are uniform appearance. Both are relatively uniform appearance. These cells are very simple, and these are prokaryotic. Okay, prokaryotic, and this bacteria sometimes are called hemotrophic, and which may digest carbon-containing compound. That's what we discussed in the BOD experiment. Okay, so which will consume energy from carbon-related compounds. That's what is called as hemotrophic. So please remember, single cell organisms. You remember size 0.1 to 10 micrometer. Okay, and which contains is one cell membrane or one cell membrane, and cell membrane or cell contains is two major regions, cytoplasm and also nucleus. And these are prokaryotic cells, and all bacteria are predominantly hemotrophic because it may digest carbon-containing compounds. Okay, yes. So based on the shape, you would also study Bacillus, Coccus, Spirillum, and Vibrio, and cluster of cocci, pair of cocci, and chain of bacilli or something. So depending upon the shape of the bacteria, you could also identify morphology, the shape of bacteria. Okay, so these are cocci varieties, and these are bacilli varieties, and these are other types. Okay, so in the water, if you go for a microbial examination, or if you go with the microscopical examination, you could be able to see these sort of pictures. Okay, so based on this, you could be able to identify whether your water is either contaminated with bacteria or other type of microorganisms. Okay, so this is the sketch of the biological cell or bacterial cell I have already mentioned to you. Okay, this contains a clear cell wall, and it also contains a cell membrane. Okay, and the cell membrane normally encloses the cell material inside, and bacteria sometimes is motile because of this flagellum. So because of this flagellum, it will also move, or you can simply call it as free living organisms. It will move here and there inside the water. Okay, so these 2 points are important, and the bacteria within the water supplies are classified into 2 groups. One is about autochthonous, another is about allothonos. Okay, autochthonous is the bacteria which emerge inside the water supply system. So please make a note on this. Something is important. Okay, two types of bacteria which are major in concern in water supply systems. Okay, which may emerge inside the water supply system. That is called as autochthonous bacteria. Or sometimes there could be a possibility of intrusion. There could be a possibilities of external intrusion of these microorganisms from outside environment. Okay, either maybe contamination or runoff or rainfall, and you can call it as allochthonous. Okay, come outside of the water supply systems. Okay, yes. So predominantly the second group of bacteria are most common in water supply systems because of your network failures, or if it is anything on the leakages, there could be a possibilities of these contamination into the water supply networks. Okay, so the bacteria which may emerge inside the water is very few, if at all anything on iron bacteria or sulfate reducing or sulfate oxidizing bacteria. So we can discuss later what it is. Okay, so there could be a possibility of emerge inside, that is because of iron bacteria or sulfate reducing bacteria or sulfate oxidizing bacteria, which emerge inside the bacterial system or which emerges inside the your water supply system. But bacteria which comes out of your network or which may intrude in the systems, that is because of contamination and surface runoff, rainfall, because of faulty joints or leakages. Okay, yes. And the predominant most common bacteria of concerns are coliforms. Please make a note on this. Something is important. Okay, if the water it is contaminated with bacterial infections, and normally they may not detect what type of bacteria, but they may detect coliforms. Okay, so this total coliform analysis have been traditionally used as an indicator of fecal coliform. Please make a note on this. Something is important. If you give a sample to the say example laboratory, and they may, and if you say if it is a contamination, microbial contamination, they may go for a detection of coliforms only, but they may not detect what type of bacterial infection it is. They may check whether the coliforms are present or not. If the coliforms are present, then there could be a suspicion that water is contaminated with some sort of microbial infections. Okay, so please make a note on coliform bacteria is something is important. Coliform bacteria, and there could be a limit that we'll discuss later. And you could expect the limit is if it is excess of 1 colony per 100 ml. Okay, 1 colony forming unit, 1 CFU per 100 ml. Okay, please remember this is the limit. Okay, 1 CFU. CFU is colony forming unit. That I'll come back later what it is. Colony forming unit per 100 ml of water indicates a possible source of any fecal contamination. Fecal contamination may be from either animal or from a human. Okay, any fecal contamination will ends up with the generation of or production of coliforms in the water, and the coliform detection is very easy. If coliforms are present in the water, you could say that water is contaminated with some sort of bacterial or other microbial contamination. Okay, and I've mentioned already iron bacteria. I'm already familiar because if it is iron rich iron in the water, there could be a possibilities of iron bacteria. So this iron bacteria oxidize Fe2 plus. Okay, normally Fe2 plus into Fe3 plus. Okay, please make a note on this. Iron bacteria oxidize ferrous ion to ferric ion and affect the precipitation of ferrihydrate. Okay, that is something is important. Okay, if more say example these iron bacteria are either aerobic or these are filamental bacteria. So please make a note on if it is any aerobic environment, if sufficient Fe2 plus are present, if any iron bacteria are existence in the pipe network, there could be a possibilities of conversion of Fe2 plus into Fe3 plus, thereby which will form a brown rusty precipitate like as ferrihydrate. Okay, so this is one example where you can have iron bacteria inside the pipe networks. It is a filamentous. That's what I told you. You could able to see now the filamental structure, and which will consume or convert Fe2 plus into Fe3 plus. Thereby, the water becomes this so turbid yellow color, or it may form a ferrihydrate as a precipitate. Okay. Yes. The second type of bacteria is sulfate oxidizing bacteria, and there could be a sulfate reducing bacteria also. Okay, sulfate oxidizing in the sense is the ability to oxidize sulfur or sulfide into sulfates. So please make a note on this point is important. Okay, this is SOB bacteria, sulfate oxidizing bacteria, ability to oxidize sulfur or sulfide to sulfate, and there could be a the sulfate reducing bacteria also, sulfate reducing bacteria. Okay, and these are anaerobic, anaerobes. Okay, please make a difference on this is aerobic and this is anaerobic, and what will happen, which may reduce sulfate to sulfide, reverse process. Okay, so please remember the two concepts, sulfate oxidizing bacteria and sulfate reducing bacteria. Sulfate oxidizing bacteria thus which have ability to oxidize sulfur or sulfide to sulfate. Okay, and if it is more sulfate in the water, probably you are already familiar, it will lead to a lot of corrosion. Sulfate, if it is more sulfate in the water, it lead to corrosion in the pipe networks. And you have a sulfate reducing bacteria in the anaerobic environment. Okay, which will be taken, or these SRBs will consume sulfate, or which will reduce the sulfates to sulfide, which is reduced to sulfide. This could also these top type of reactions also may happen inside the pipe networks. Okay, and nitrifying bacteria, anyway I'm not going to discuss. Nitrifying, we are already familiar, Nitrobacter and also Nitrosomonas. These two bacteria are important in concern in the water supply systems, but these are strictly aerobes. Aerobic condition will work out, but these are not pathogens. These are not disease causing organisms. Okay, so Nitrobacter role you are already familiar, organic nitrogen and nitrogen hydrolysis into NH4 plus, NH4 plus into NO2 minus, nitrites by Nitrobacter, further into nitrate by Nitrosomonas and also Nitrobacter. Just make a note on how the conversion of organic nitrogen into nitrates. Okay, yes. Yeah, the second major group of microorganisms are protozoa. Protozoa, these are higher living organisms. Okay, than your bacteria. These are unicellular and again animal-like single-cell organisms, non-photosynthetic and non-photosynthetics, and protozoas are highly motile, which will also move, and because of this flagella. Okay, and you can also have amoeboid locomotion or by cilia, that also you would have studied, and these are the different representation of protozoas, either amoeboid protozoa or flagellated protozoa or ciliate protozoa. So you could also see different types of amoeba, it is one example, ciliates is other example, flagellates is another example, Suctoria and sporozoa. So please remember these are different groups of protozoa in water: amoeba, ciliates, flagellates, Suctoria, and sporozoa. And these are some examples: amoeboid, flagellates, and also ciliates. And you would have also studied or something like noticed in either biology or some water chemistry. Okay, so protozoa of concern in water. Okay, these important characteristics of protozoans are this is larger than bacteria, so that's what we discussed. Up to 10 micrometer is bacteria, this is 10 to 15 micrometer, and this protozoa contains specialized organs for feeding, digestion, and locomotion. And hemoheterotrophic, please make a note on this is hemo again, which will consume carbon, and this is from dead organic matter. Okay, please make a note on hemoheterotrophic. Bacteria, what we have discussed, is hemoautotrophic. That is hemotrophic organisms. Here, hemoheterotrophic organisms. Okay, and most are aerobic and or facultative anaerobes. Facultative aerobes, maybe strict aerobes, or it may be either facultative condition. And these protozoas are highly motile, or it may have free swimming by cilia or propulsion. So that also you can make a note on. And a drawback with protozoa is, last point, if any favorable conditions present, it will grow in number, and if it is any unfavorable conditions exist in the water or in the system, it may form a protective coating and stay for a long period of time, and it will also reproduce once the comfortable environment exists. That something is important. These protozoas are highly vulnerable. Okay, and that's what If it is anything on a favorable environment, it will grow in number. If it is anything on unfavorable conditions, it will form a protective coating. Okay, it will be there in the system within the coat and live for a long period of time. And if condition prevails, it will also reproduce further. Okay. Third composition, algae, predominantly in the water. You would have come across algae or aquatic plants. And these can be seen either by naked eye or by microscopic analysis. Okay, it's very common in, you would have seen in lakes and also reservoirs, and this water turned to be in green in color. That is because of algal, suspended algae. Okay, and these algae occur in fresh as well as polluted water. Okay, so if it is anything on nitrogen and phosphate enrichment in the water, and there could be a production of algae. So most of the Kerala, I would have seen all the ponds are green in color. That is because of excess algae production. Okay, and these are plant varieties. Okay, and which will have a capability of using energy. Okay, it will tap energy from the sunlight, and it will also undergo a process called photosynthesis. Okay, these are photosynthetic plants. Okay, yes, and these are common algae present in the water. Green color algae, euglenoids, and diatoms, and cyanobacterium. At least you remember the names of major algae present in the water. Green algae, which contains a lot of chlorophyll. This is very common. And euglenoids, these are also again single-celled green pigmented algae. And that also resembles something like a protozoa. Diatoms you would have come across. These are golden brown, single-celled, hard cilia shell type algae, diatoms. And last one is about bluish green in color, cyanobacterium, which also undergoes photosynthesis. These are otherwise called as blue-green algae. Okay, please make a note on cyanobacterium. These are 4 or 5 different types of algae present in the water. So this situation you would have seen in surface waters most commonly. Okay, and green color, that is because of suspended algae. Okay, you could also seen in surface water sources like lakes and also ponds. Okay, some sort of green in color because of suspended algae. Okay, last one is about virus. Okay, and please remember, viruses are intracellular parasites, and they do not have independent metabolic and reproductive system. Please make a note on virus is not being considered in water treatment system because virus doesn't have a metabolic activity. Okay, it will not have a metabolic activity. Okay, and it may live on only the coast. Okay, it will live on only the coast. Okay, so virus doesn't have a virus cells. There are no virus cells, and virus cells are simple proteins, virions, which are the strands of genetic material contained within the protein coats. It's very common. Please remember, virus cells are simpler protein coats. Okay, these are intracellular parasites. These are parasites. This does not have any metabolic or reproductive system. Virus can infect human, animal, plant, bacteria, fungi, and also algae. And these are the size ranges from 10 to 25 nanometer. Please remember the size range. Okay, 10 to 25 nanometer. This is the virus cell. Anyway, you just need not. And the viruses are very dangerous. Probably would have come across. Okay, imagine our situations. One cell is infected with one virus attack. Okay, so that we can call it as a dormancy. Okay, dormancy is number 1. A, dormancy, and B, and the virus cell it is getting soaked onto a surface of a single cell. Okay, that you can call it as adsorption, and penetrate inside the cell, damages the cell, and grow in number. And because of this viral exposure, the single cells will be blasted into. And now just imagine one is entered in, and you may get 1, 2, 3, 4, 5, 6 multi number. It will also infect multi number of cells thereby, and in a short period of time there could be a complete viral exposure. That's what people used to say. It's very, very difficult to control the growth of virus once it is infected. That's what now people have a concern in virus in water. Okay, yes, and this virus I already mentioned to you. Virus consists of core nucleic acid surrounded by a protein coat. Okay, And because of the nanometer size range, last point, virus could not be detected without an electron microscope. Normal microscopes are not in a position to view our virus traces. So you need to have electron microscope to view your virus in the water. Okay. So normal virus you would have come across. Enteric viruses of particular concern in waters are hepatitis is very common. Hepatitis virus, okay, and rotavirus you would have come across. Adenovirus you would have come across. Enteroviruses you have come across, and the last one is about reoviruses. Please make a note on. And these are important particular concern in water. Important enteric viruses of particular concern in water. Okay, hepatitis is very common. You may get hepatitis virus, hepatitis, infectious hepatitis, rotavirus, which may also give sort of gastrointestinal problems, especially with the children. Adenoviruses normally it may infect your intestine as well as your upper respiratory tract. Third one, an enterovirus, which will affect both intestine and respiratory tract, and reovirus again, which also infect both intestine and upper respiratory tract. That you just have a look on. Okay, and some of important pathogenic viruses or the microorganisms are concerned, you would have already come across these diseases. Typhoid is by Salmonella typhosa. Please make a note on this. Infection is important. Infection name and the organisms or bacteria which is responsible for. Okay, and you may get a dysentery, very common, and you may get dysentery because of Shigella flexneri, a particular organism. Okay, and tetanus you would have come across, that is by Clostridium tetani. And botulism you would have come across, that is by Clostridium botulinum. That's a virus. Okay, and diphtheria, that you can have pathogenic organism is called Corynebacterium. So please remember, you are something is important, and infections, okay, and type of microorganisms. So you are in a position to say typhoid by Salmonella typhi, dysentery by Shigella, and tetanus by tetani, and botulism by botulinum, and diphtheria is by Corynebacterium. So please make a note on this. Is something is important. These are waterborne diseases. Okay. Yes. So I'll stop here, and now being familiar with these microorganisms, now you have to analyze the water, whether it is contaminated with bacteria or not. Okay, that's the last portion that we'll cover it tomorrow. Thereby we'll close physical, chemical, and biological or microbial characteristics of water. Is it okay? Is it fine? Anything to discuss or any clarifications? Because whatever we have discussed today is about COD is very simple. Chemical oxygen demand. You just use strong oxidizing agents to oxidize the organic matter into oxidized product. You may use potassium dichromate as a strong oxidizing agent in an acidic medium. You may also use silver sulfate as a say example catalyst. In an elevated temperature, you have to digest the sample. After digestion, you can do it either in a closed reflex method by titration. You can estimate the COD of the sample by simple titration methods against ferrous ammonium sulfate using ferrion as an indicator, or you can digest in a digester, CO digester, and you measure the absorbance in the spectrophotometer. That's what we discussed. Okay, and the next portion what we have seen today are microbial contamination or microbial characteristics of the water. And we started with bacteria. Second one is about protozoa. Third one is about the algae, and fourth one is about virus. And please be familiar on at least 4 or 5 important characteristics of each and every item, and what type of organism it is. Okay, either chemotrophic or autotrophic or photosynthetic organisms, or parasite or pathogen or something. So just to remember these concepts in one or at least 4 or 5 points. And you can last point what we discussed was is number bacteria or pathogens of concern in the water treatment. Okay, typhoid, that what we discussed, dysentery by Shigella, and tetanus, botulism, and last one is about so please at least read and have some hint for the examination point of view. Is it okay? Is it fine? Rashwant?
Okay, sir.
Yes, sir.
Yeah. Yeah, thank you. Just have a look. But lectures are lengthy, but all of the things are not necessary for the examination. You just take out what is this your requirement for your examination point of view. Just see if this is soft copy way of reading, you just highlight the points, capture it, or just take a note or bit of paper and write one by one and refer it for the examination point of view. Because if I ask physical characteristics or chemical characteristics, and it is somewhat difficult to write a lot of stories. Okay, so just concise and read, prioritize your answers, and based on that, you can also either MCQs also sometimes is easy to answer, or some sort of descriptive questions also is easy to answer by yourself. It's fine.
Yes, sir.
Yeah, thank you, Rashwan. Thank you all. Yeah, bye.
Bye, sir.
Bye. Yeah, thank you.